All right, so we're going to take a look at chapter number 18, and chapter number 18 is looking at our introduction into the nervous system. So we are specifically going to talk about organization of the nervous system and also the cells that we see incorporated with the nervous system overall. So first up, general function of your nervous system is ultimately going to be communication. Nervous system really doesn't work alone. It works hand in hand with another system, and that system is the endocrine system. Because they often work so closely together, it is commonly referred to as your neuroendocrine system. Remember, endocrine system is going to include all of the endocrine glands and the hormones that they secrete. So looking at the thyroid gland, pituitary gland, adrenal glands, and so on. Overall function, again, is communication. So communication within part of the body or communication from one part of the body to another and so on. Okay, so it's all about communication. If we see what we find within the nervous system, we are going to find a few components. And the way we organize these components, we can actually look at this picture right here. And we can see we will divide it into two basic divisions for the nervous system. First off, we have our central nervous system, which we commonly just call CNS. And then we have our peripheral nervous system, PNS. As far as the CNS is concerned, it's everything that you see here in green. So your CNS, your central nervous system, solely is comprised of the brain and the spinal cord. Those are your two components. Is there anything else? No. Your central nervous system is just the brain and just the spinal cord. This is the central portion of your body, so the central portion of this nervous system. This is like the core of the nervous system. Now, as far as what we have in yellow, that was our peripheral again, and we can see everything in yellow are the nerves of the body. With the nerves of the body, we have the cranial nerves. There are a dozen pairs of cranial nerves, and then we have the spinal nerves. There are 31 pairs of spinal nerves. And if I hop back here, okay, again, we can see nervous system is made up of the brain, the spinal cord, and the nerves. So yet, the spinal cord and the brain are both central nervous system, and then, of course, the nerves are peripheral. So purpose of this system overall, again, is going to be communication, and here we can see exactly what is going to take place. So we need to be able to detect changes in both the internal and external environment. So we need to be able to experience if we are placing our hand on something hot or something cold, or if there is something painful. So we have to be able to detect these changes. Not only that, we need to evaluate this information. And we need to come up with a plan. So we initiate an appropriate response. So I place my hand on something really hot, and ultimately my arm is going to kind of retract my hand from that hot object. Okay, so picking up on these changes, evaluating the information, and then coming up with a plan. Here we see a nice little review of central versus peripheral nervous system. Central, again, this is your center of your entire nervous system, and all we find is just the brain and the spinal cord. Okay, and this is kind of like the big boss of your nervous system. This is where all the integration and evaluation and coming up with this plan takes place. Peripheral nervous system, on the other hand, is just made up of those nerves, and the purpose of those nerves is just to send that information to the central nervous system or to carry the information away. There is no integration or evaluation or coming up with a plan. They are just there to carry that info either towards the central nervous system or away from the central nervous system. And again, we find cranial nerves and we find spinal nerves. Cranial nerves are 12 pairs. You do have a cranial nerve assignment that you need to complete. Instructions are on Blackboard. But we do have 12 cranial nerve pairs, okay? And with these pairs of cranial nerves, so they're going to originate from the brain. Some of them are sensory, and sensory means they carry information towards the brain. Others are motor, and that means they carry information away from the brain. And then some of them are mixed, meaning they have fibers that carry information towards the brain, and they have fibers that carry information away from the brain. Okay, but bottom line, your cranial nerves are associated with the brain. You have 12 pairs, some sensory, some motor, and some of them are mixed, meaning they have fibers from both sensory and motor. Now, as far as your spinal nerves are concerned, these are going to originate from the spinal cord. And as you can see up here, we do have 31 pairs of these spinal nerves. So these 31 pairs, they're all mixed nerves. That means they carry sensory information, and they also can carry motor information. So they carry information towards the spinal cord, and they also carry information away from the spinal cord. Naming the spinal nerves is pretty straightforward. They are named after the vertebrae that they're associated with. The only odd thing about this is there are 8 cervical spinal nerves. There's only 7 cervical vertebrae, but there are 8 cervical spinal nerves. Okay, so you have that additional spinal nerve, and then the numbers are consistent all the way down the column, and we do have one coccygeal spinal nerve as well, one pair. Okay, but they're all mixed yet again. And you'll have to name them in a lab that is due for our next exam. This picture is absolutely fantastic. I really like this picture. It's right out of your book. And it lays out the complete organization of the nervous system. So if we look at this, we can see a few things right off the bat that you guys already know. First off, central nervous system is everything up here in this tan box. Okay, so up here, this is your brain and your spinal cord. Peripheral nervous system is all down here. So peripheral, yet again, is going to carry information up towards the CNS or down from the CNS. Okay, so again, like we talked about before, motor information is carried away from the CNS, and sensory information goes towards the CNS. Now, I can put a little drawing up here for you. Let's do this real quick. What we have up here is our CNS. Okay, so this is our brain and our spinal cord. This is all up here in the CNS. So that'd be like the tan box in that picture from your book. And I'll use the same colors that they use in your chapter that we have in our notes. And you're going to see a blue arrow coming up towards the CNS, and the blue arrow is going to represent what we call sensory. Sensory is also known as afferent with an A. Okay, so sensory afferent goes towards the CNS. Okay, so we can say towards. So they carry incoming information towards the CNS. On the flip side, moving away from the CNS, and I'll use red to kind of show the red in the book. This is motor. This is efferent with an E, so carrying information is going to move away from the central nervous system. Okay, so this one we're moving we're moving away from the central nervous system. Now down here, okay, below the sensory, this is where we're going to have whomever picks up on the sensation. So what picks up on the sensation is some type of a receptor. There's a stimulus, like me placing my hand on something hot. Well, I have temperature receptors in my hand. They're going to pick up on that, and they're going to send the information to the brain and say, okay, hey, Lori, that's really hot. Here's what we're going to do about it. They come up with a plan, and then the CNS sends the plan out efferently with these motor fibers, and it's going to go to some type of an effector. And the effector is where the response happens. And in that scenario, if I place my hand on something hot, what's going to happen is my hand's going to retract from that hot object and I'll remove my hand. Okay, so this is kind of your general look at sensory versus motor. This right here, if we were to get all technical and kind of name this, we would say this is a two neuron reflex arc, because there's two neurons that I drew, okay, coming in afferently and moving out efferently. Okay, so now let's look at some more details with our presentation. Okay, so here, when we look at this, we can see that there's two different types, or two other divisions of the nervous system. We have a somatic and we have an autonomic. Difference between the two is going to be the type of effectors involved. Okay, so if we think back to the two neuron reflex arc picture we just drew in our notes that I drew on the board, the effector is where the response takes place. Okay, so with the somatic nervous system, soma means body. Okay, so your somatic effectors are skeletal muscle. Okay, so with the somatic nervous system, your effectors are skeletal muscle. And if we look at this picture, this is just like what we drew on the board. You have some type of a receptor. You're going to use that blue arrow, which is afferent. So afferent is sensory. Carry it up to the CNS, come up with a plan, send that information out with the pink or the red one, which is motor or efferent, and we're going to carry it to some type of an effector. Because it's the somatic nervous system, our effector is skeletal muscle. Okay, so soma means body. Skeletal muscles are the muscles that make up the form and contour of your body. Okay, so remember, we do have willed control over these skeletal muscles. Now, on the other hand, if we look at what we got here, we have the autonomic nervous system. Autonomic nervous system, I'd like you to think of automatic, okay, because it is automatic with what's going to happen. We don't have control over these effectors. So first up, we can see it's kind of starting the same way, where we have our receptor, we carry it up afferently, sensory division, up to the CNS. And then we can see we have two arrows, but bottom line, we're still carrying it away efferently. And we're going to go down to some type of an effector. Now, with your autonomic nervous system, your effectors include cardiac and smooth muscle, which we know we don't have control over. Also our glands, we can't tell our glands when to secrete. Also other types of tissues. So these are all automatic, autonomic effectors. So that's the difference, between your autonomic nervous system would be these types of effectors, and then your somatic nervous system, which is skeletal muscle effectors. Now, what in the world are these two pink lines? Like, why are there two divisions that are efferents, that are leaving the central nervous system? Well, we have what we call our sympathetic division of our autonomic nervous system and our parasympathetic division of our autonomic nervous system. Sympathetic division is involved with what we call our fight or flight response. This happens when there's some type of a threat to you. So a really good example would be you're hanging out outside at some park and then all of a sudden a dog starts running towards you and is barking and you become alarmed. So this is a threat to you. So your body is going to do some things. There's going to be some changes that take place in order for you to, heighten your awareness and be able to either fight. So yeah, you're going to fight the dog, or take flight and flee the situation, get out of there. So you're going to start running really fast, or you're just going to get ready to fight this animal. So of course, blood flow is going to alter. We need to, deliver some extra blood to our skeletal muscles so we can run even faster. We're going to see that our heart's going to start beating more quickly because you're getting nervous. We need to get that blood going. The rate of breathing is going to alter. You're going to notice there's a change with your pupils as we need to just kind of heighten our awareness and kind of get as much visual stimulation as we can. These are all sympathetic type changes. Now, parasympathetic, this is what we call our rest and repair or rest and digest, normal activities division. Okay, so now the threat is over and you're relaxing, everything is going back to normal. You safely made it away from this animal. So, your heart rate's going to decrease back down to normal. Your normal breathing is going to resume. Your pupils are no longer as dilated as they were, so on and so forth. Okay, so now your body can do things like continue digesting your lunch. During the threat, your sympathetic division stops the digestion so your body can prepare for other events that might happen. Normal activities, this is when we resume digestion and just go on with our regular day-to-day normal, biological activities. Okay, so again, big differences with the effectors. So if we look at this picture on the board again, over here, what we can do is we can look at these effectors and we can say, SNS, which stands for our somatic nervous system. The effectors are skeletal, would be skeletal muscle. And then the effectors, if we look at the ANS, which is autonomic, the effectors would be cardiac and smooth. These are both muscle. And your glands. Okay, and of course other types of tissues too. But this is kind of your big difference with the effectors. So depending on the effector type, or what division of the nervous system to look at. So really good picture that is right in your textbook. Little review of everything that we just talked about. So here we can see afferent, again, is all incoming information, all sensory, all going towards the CNS, where efferent division is all outgoing or motor or moving away from the central nervous system and going towards some type of an effector. Somatic nervous system, we are looking at our effectors being skeletal muscles, okay? And then we look at the autonomic nervous system. Again, we are looking at our effectors being glands, smooth muscle, and cardiac muscle. And of course, with the autonomic, this automatic division, we have two subdivisions, either sympathetic, which is fight or flight. We need to, fight some situation or run away from the situation so your body prepares for those events. And then parasympathetic, this is when we kind of go back to normal, so our regular resting activities. Again, you can call it rest and repair, or sometimes rest and digest. So that is the difference between the two. Your response comes from different places along the central nervous system. Like, for example, parasympathetic is oftentimes referred to as the craniosacral division, because you're looking at, the upper and lowermost portion of the central nervous system, where sympathetic, this is your thoracic lumbar region of the spinal cord. that is more responsible for these types of responses. Okay, so it's kind of like the middle part of the central nervous system. So functionally, anatomically, there is a difference between the two. All right, so next up we're going to talk about cells. And with cells, first off, we have our general nerve cells. Okay, so our nerve cells are called neurons. Those are just our general cells. And then we have what's known as our neuroglia. So these are all of the little tiny support cells which surround your nerve cells. If you remember back when we talked about the tissues, we looked at having the nerve cell, and then I put all these little speckles, all these little dots, and I said these are the neuroglia, these are the glia, and they surround the nerve cells. Okay, and they totally outnumber the nerve cells as well. We can see up here that there are 900 billion of them, so a ridiculous amount of them. Now, with these neuroglia or glia cells or glial cells, and again we call them neuroglia, so that'd be Neuroglia, neuroglia. Okay, the term glia means glue. So they first thought when they were discovered that they just kind of held everything together like glue, but soon we realized, well, yeah, there's 900 billion of them, but there's 5 types. Okay, so there's 5 main types, and each of the 5 main types actually does something different. Okay, bottom line, they do provide support, as you can see up here. But let's talk about the differences between the 5 types, because I'd like you to know this stuff. All right, so type number 1, we can see right here, is called an astrocyte. Astro basically means it's shaped like a star. Okay, so it's a cell that's shaped like a star. So they are star-shaped, they are the biggest, okay, so they're the largest, and by far they are the most popular, the most numerous type of glial cell that we have. They're kind of cool with what they do. If you look at this picture, here we can see, this is a capillary. Okay, so we have blood flowing through the capillary. You can see that these astrocytes, they actually wrap around the capillary. And then the other end, they don't really show it in this picture, but the other end of the astrocytes, they connect to a nerve cell. Okay, to a neuron or a nerve cell. So if my arm was the nerve cell, it would connect to my arm, which is the nerve cell, and then of course the blood vessel, so the capillary. And these astrocytes, what they do is they are going to allow nutrients to travel from the bloodstream through the astrocyte over to the nerve cell. Nerve cell ultimately has waste, and the waste can travel through the astrocyte back into the bloodstream. Okay, so they're kind of like that transportation pathway between the bloodstream and the nerve cells. Okay, so here we can see they connect to both nerve cells and capillaries, and they allow this nutrient transfer and waste transfer. So very important, they allow that, but not only that, they're pretty strict with what can actually leave the blood and get into the nerve cell. Okay, so we see there's tight junctions. We know tight junctions, not a lot gets through, but it actually makes up what we call the blood-brain barrier. That's what BBB stands for, blood-brain barrier. So it is the barrier between the brain, well, the central nervous system, and the blood. So they can control what can leave the blood and get to the brain or to the central nervous system and what can't. Very important because there's, some different chemicals and whatnot that are in the bloodstream that we do not want to have access to our central nervous system. So blood-brain barrier formation, these astrocytes are, take on a leading role with that. Also, you'll notice CNS in parentheses, and that means that you find the astrocytes in the CNS, so we find them in the brain and we find them in the spinal cord. Okay, we don't find them associated with the PNS, so with the CNS. In fact, I told you there's 5 types of glia we need to talk about. 4 out of the 5 types are associated with the CNS. There's just 1 type, which is PNS only. All right, so the next one that we have. Next one that we have are the microglia, okay, or you can think of it as microglia. These 2 are central nervous system. Micro, I'd like you to think small, and that's what they are. They're tiny. Okay, so they're small little, glia, and what they do is, honestly, they don't do much. Day to day, they're not very active. Okay, so you can see up here, they're usually stationary, so they're really not doing too much. The only time they become active and actually do anything is if there is inflammation within the central nervous system. If there is inflammation, these microglia can grow in size and become active and start to break down components. Okay, and they undergo what's called phagocytosis, and phagocytosis is simply engulfing and destruction of molecules. So if there is something foreign present, and here's the the microglia, and what they can do is become active. They can go towards the foreign entity, engulf it, kill it, destroy it, and that's what phagocytosis is. So engulfing and destruction of material. So they get bigger, become active when there is inflammation. Otherwise, they're just tiny and they're really not doing much. Next one we call ependymal. And ependymal, hopefully, first thing that pops into your head is epithelial, because they look like epithelial cells and they kind of act like epithelial cells too. So these are associated with the central nervous system. Okay. And if you look at them, they really do look like epithelial cells, because they kind of look like, elongated cuboidal cells with some cilia on them. And if you remember what cilia does, cilia is going to kind of sweep material across the surface, and that's what they do. So you find these cells with their cilia lining the fluid-filled areas within the central nervous system. Okay, so fluid-filled areas would be like the ventricles of the brain. Okay, we have 4 ventricles of the brain that we will see in the next chapter. And these 4 ventricles of the brain are filled with fluid. This fluid also travels through the central canal of the spinal cord, and we also find it in the subarachnoid space. So this fluid is actually moved around by these ependymal cells. The fluid is never stationary. It is constantly circulating. That's why they have the cilia, because the cilia is going to keep the fluid moving. Now, very interestingly, these cells, not only do they keep the fluid moving, but we can see here, some of them actually produce the fluid. So they exist in little packets, and we'll talk about what these packets are in a later chapter. But these little packets, they're called choroid plexus, but what they do is they produce this fluid, and this fluid has a name. The name of the fluid is CSF, which is cerebrospinal fluid. So I'll just kind of put that on the board so you can see how it's spelled. So I'll just move this on over. Let's see. So we have CSF, which is cerebrospinal fluid. Okay, so these ependymal cells are going to produce this fluid, and they're going to keep it moving. So they're going to keep it circulating throughout the central nervous system. And we'll talk more about this in the next chapter as well. Okay, so let's go back to our presentation. And so we just finished number 3, which are the ependymal cells. So next up, we'll take a look at number 4. These are called oligodendrocytes. It's kind of a mouthful, the name, but if we break it down, oligo means few. Okay, so there's not a lot, so just few. Dendro, think dendrite, so like extensions. Cyte means cell. So there's cell that has few extensions. If we go back to the astrocytes, let's kind of hop back here. Astrocytes, you can see, have lots and lots of extensions sticking out of them, where the oligodendrocytes, by name, by definition, just have a few. So if we look at this oligodendrocyte, it's got one here, and one here. So just a few. So they're smaller than the astrocytes and they have fewer processes sticking out of them. So what they do is they're going to hold together nerve fibers. They're going to hold them all together and ultimately form what we call the myelin sheath. Okay, so this myelin sheath is an insulating sheath for these nerve fibers. Nerve fibers that have a myelin sheath allow information to travel much more quickly. Okay, so if they have the myelin sheath, information travels faster than if they don't. So here we can see this fiber, this extension, reaches out and just wraps around the blue nerve fiber many times, and that's called a myelin sheath. So it provides that insulation and helps speed up the rate of transmission of information. We zoom in a little bit and look at this myelin sheath. Here's a nerve fiber. Okay, so it's an axon. An axon is a nerve fiber. And then we see that this myelin sheath, we have these cells that wrap around. This picture is showing what we call a Schwann cell, but these oligodendrocytes do the same thing. Okay, so it's going to wrap around and form that myelin sheath. So let's talk about these Schwann cells. So oligodendrocytes are in the CNS. Okay, so central nervous system has the oligodendrocytes, and then, like I mentioned, the Schwann cells, these are in the peripheral nervous system. These are the only type of glial cell which you find in the PNS. They do the same thing as the oligodendrocytes. The only difference is Schwann cells are in the PNS, oligodendrocytes are in the CNS. But they're both going to support the nerve fibers and wrap around them and make up this myelin sheath. Okay. There's a few other definitions up here. One thing to keep in mind, these cells typically cover the fibers. Okay. If these cells decide to cover the cell body, then we call them satellite cells. But if they cover the fibers like the axons, then we call them Schwann cells. But if they cover the cell body, then they're satellite cells. So if we look at this right here, okay, so here we can see the myelin sheath around the nerve fiber, and then we can say this nerve fiber is ultimately myelinated because it has that sheath around it. So this is the fiber, okay, that has the myelin sheath. Okay, so neurolemmacyte is the cell which wraps around. And then here, the cell body, if it's wrapped with this sheath, it is called a satellite cell. Okay, it's just the terminology is a little bit different, but it's the same thing. Okay, same fundamental thing. Let's go back here. Okay, so neurolemma. Okay, so that's basically what you're going to have when you wrap that cytoplasm all the way around. And then the Schwann cell is sometimes called the neurolemmacyte, which we saw in this picture right here. Okay, so that's going to wrap around the fibers or the axons, and the satellite cell is what attaches to the body. Now when we have this, I'm going to draw you another picture. Let's go back to our board here. Let's turn on the light as well. Okay, so here's our nerve fiber. Right now it's unmyelinated, so let's add some myelination. So what I'm going to do is I'm going to add this myelination like so. Let's color it in. So here's some myelination. Here's the next myelination. Okay, so it's going to exist like in these blocks, like so. Okay, and there's a gap in between these blocks. So we have that myelination. Now with these little gaps These little gaps have a name. Okay. So nodes of Ranvier, or technically it's called nodes of Ranvier, but so these little nodes are little gaps in between, and what they do is they are going to, that marker doesn't work. They allow something called saltatory conduction, which you'll learn all about in advanced A&P, but that basically allows information to travel very, very quickly, and the information pretty much hops from node to node to node much more quickly than it would if it passed through completely. Okay, so kind of leapfrogging like this is called saltatory conduction, and it's very, very rapid. So we can say it is rapid. Rule of thumb that we have is this. Thicker is quicker. So if you have that myelination, you see that the nerve fiber has become much more thick. The thicker it is, the quicker the information can go. Okay, so the faster it is, the thicker it is, the faster the information can travel. Now there is a condition which you have, if you have breakdown of this, so let's say this is starting to break down, and when it breaks down, it kind of forms this scar, and this is what's referred to as a sclera, and that's going to kind of interfere with transmission of this information. So if this happens lots of times, okay, so we can do some and just put them there. So if you have lots of them, you have what we would call multiple sclera. And this condition would be called MS, which is multiple sclerosis. Okay, and again, sclerosis, of course I forgot the L. Okay, multiple sclerosis is because there's multiple sclera. Okay, so that would be MS when you have breakdown of that myelin sheath. Also, one more thing we can see if we look at, if we go back to, let's go back, there we go. If we go back here, we can see that when you do have myelination, we call these fibers white fibers. And when we don't have myelination, we call them gray fibers. So white fibers, or AKA white matter within the brain and spinal cord, is myelinated. Gray matter within the brain and spinal cord is not myelinated. Okay, so that would be unmyelinated tissue. Okay, and then next up, we have what we call a neuron, and I think I'm going to do all of this up here on the board. So we're going to go ahead and just draw a picture of a neuron like we have done before when we talked about nervous system tissue. So we have, this is what we would call a neuron. Or a nerve cell. Okay, so that's a neuron or a nerve cell. A few different components that we have with this nerve cell. We have what we would call, and I'll just kind of dot this in green. This is our cell body, also known as a perikaryon. P-E-R-I means to surround, kary means the nucleus, so it's the part of the cell that surrounds the nucleus. So in here you have a nucleus, you have ER, you have mitochondria, you have all the regular organelles like any other cell. So there's your typical cell body. What makes a neuron or a nerve cell unique are all these extra little things. Okay, so now I'm just going to use orange for these little pointy guys. Okay, so all the way around right here, these little pointy guys, these are what we call dendrites. Okay, so these dendrites, let's see if I can zoom in for you guys a little bit. Okay, so we can see dendrites. They carry information towards the cell body. So technically, I guess I'll just flip the arrow. Okay, so they carry information in. I could do this too. Let's just do it this way. So they carry all the info in. That's incoming. So dendrites carry info in towards the cell body. And then in blue, we have this guy. This is what's known as our axon. And our axon would carry info away. Carries information away from the cell body. So information is traveling this way. Okay, so those are our three primary components that we see with a basic nerve cell. So again, the cell body has everything within it. Now, what's going to happen at the end? Let's just make this a little bit bigger. This is going to be totally out of scale, but what we see here, see, they kind of look almost like little doorknobs. Okay, so at the end here, we're going to have what we call a synapse. Okay, a synapse is when you have the information leaving one neuron, connecting to another neuron, or connecting to a muscle or a gland or something. So let's put this guy here. This is going to be a muscle, let's just say. Okay, and then we have this synapse, which is going to happen. This junction is called the synaptic cleft. This thing that looks like a doorknob is called the synaptic knob, and then the synapse is basically what's going to happen here. So when one neuron talks to another neuron, or one neuron talks to a muscle, it's going to release these little chemicals. So these little chemicals that get released are called neurotransmitters. Okay, so they basically carry chemical info from one neuron to another, or to an effector. Okay, so it's going to release this neurotransmitter. So you have in the perikaryon, you have ribosomes that make proteins. Proteins can be these neurotransmitters, and they get sent down the axon. They're hanging out here. When they get the signal, they get released. They're going to bind to a receptor on the muscle, and the muscle is going to know whether it should contract or whether it should not contract. Okay, so that's what we're looking at as our little, for chemical info, we can say, I think signals is a good word. It's a little chemical signals. Let's go back to our lecture and recap a lot of this. And then we're going to take a little break and then resume with part 2 of this chapter. So neurons, once again, these are your nerve cells. Okay, different components. We have the cell body, which we call the perikaryon that we drew on the board. And that's where we find all the organelles, as you can see here. These protein constructed neurotransmitters, okay, allow the information to travel. Of course, there's mitochondria for energy. That's kind of a given. The cell needs energy to do things. So if you look at this picture in your book, we can see this right here is your cell body. It just looks like any other cell that we have talked about. And then we have all these little extensions, which are the dendrites. So if we look at dendrites in more detail, there might be one dendrite, there might be more than one dendrite. Typically, we see multiple dendrites. And again, they carry information towards the cell body, so to the cell body. Distal ends of dendrites are receptors. There's a receptor on the end that picks up on the information and it sends it to the cell body. The axon is that one single, really long process, okay, which is going to leave the cell body, and that carries information away. I'll talk to you about the axon hillock in a minute, okay, but it's going to carry information away from that cell body. And then the telodendria are a bunch of synaptic knobs together, and the ultimate distal end is that synaptic knob. Length and diameter vary, so some are really long and some are super short. It just kind of depends on which axon we're looking at, and of course diameter can vary if they're myelinated or not, and again, thicker is quicker, so if they have the myelination, they're pretty thick. So here we're looking at that myelination, but I want to go back to this picture. So the axon hillock is that little junction right here between the cell body and the axon. So this little triangular area is the axon hillock. Here are your dendrites. They have receptors right on the ends to pick up on that information, and they carry it in towards the cell body. And this one super long axon carries the information away. And we can see that this one axon, yeah, it can branch later. That's fine. Just call them axon collateral. But we can see how long this one is. Okay, so we can see it ends there. Before we get to the end, if we look, here we can see the Schwann cell. Okay, so the Schwann cell. And we know that that's going to provide the myelination in the peripheral nervous system. You see the Schwann cells in the PNS. They kind of look like hot dog buns on the axon. These are your little nodes right here, little node of Renoir. So the information just kind of jumps these nodes. And here we go, it's labeled node of Renoir right here. And if we go all the way to the end, you can see all the little telodendria. And then if we go to the most distal end, like the little tips, those are what looks like the doorknobs, and those are referred to as the synaptic knobs. Okay, so it's just your typical neuron structure. Okay, myelinated axon, we can see here yet again. Here's your nerve fiber with the myelination around it. Axonal transport, this is just how we get information down the axon. So from the cell body all the way down the axon. So if we zoom in and look at this picture, it looks pretty crazy, but here's your cell body. They didn't put any dendrites on it just to make it more simple. But bottom line, we're gonna make some neurotransmitters, okay, up here within the cell body, and then we need to get them all the way to the synaptic knob. So the best way to do that is almost like having a moving walkway all along the axon, which is called axonal support. So you have these microtubules right here, are these green guys. So you have the microtubules, part of your cytoskeleton. Microtubules can take these vesicles, take mitochondria. Vesicles contain the neurotransmitters, as you see right here. Also mitochondria that we need some energy down at this end to do things. And we can just kind of move them all down, kind of like a moving walkway. And then down here, we can release these neurotransmitters. And then, of course, they can be taken back up again, and information can go back this way for destruction by lysosomes or whatnot. Okay, so that's checking out axonal transport. Functional regions of the neuron. We have a few different functional regions. We drew structural regions on the board already, but let's look at the functional regions. So if we look at this picture, I think I'll just explain it by showing you the picture right here. I'll kind of go back and forth, I suppose. So input zone are the dendrites in the cell body. That's all of the incoming information. So if we look at this picture, okay, input zone, this is where all the information comes in. The receptor of the dendrite, it comes in, goes towards the cell body. So all incoming comes this way. Now, all the information that comes in, not always is there a plan, okay? There can be a little bit of information coming in, but it might not be enough information to send info all the way down the axon and release neurotransmitters. So this neuron has to decide if it's important enough. So that's why we have this second zone. It's called summation. To summate means to add together. So at the axon hillock is where we add together all of the information that came in. So we're going to take some information in, take some more in, take some more in, add it all up right here. And if we hit threshold, if it's important enough, then we send the information down the axon. Okay, so summation is just where we add it all up. Conduction zone, that's along the axon. That's where we conduct all the information towards the end of the neuron. And then output zone, that's where all the information finally leaves or is output. And that would be the telodendria and, of course, the synaptic knobs. Oops, went the wrong way. So here, it's going to conduct down the axon, maybe down the axon collateral, and then ultimately reach the output zone, which telodendria to the synaptic knobs, and then it'll go to the next neuron or some type of an effector. Okay, this is where we're going to take a little break, and then I will resume this chapter with part 2.